I want you to picture something for a second. Actually, let's look at three very different scenarios. First, imagine a sled dog out in the snow.
Okay, sled dog.
Right. Most of the pack is panting. They're perfectly adapted to the freezing temperatures. But one dog in this pack is suddenly able to tolerate extreme heat. It doesn't overheat. Its body just handles the temperature spike effortlessly.
Oh, wow.
Yeah. Next, shift gears and picture a spider. It's spinning a web, but this web is drastically more flexible and stretchy than the web spun by any of the spider siblings.
Like noticeably different.
Exactly. And finally, imagine a population of Yukon River salmon. They are swimming through water temperatures that are essentially boiling by their standards. I mean, temperatures that would normally be instantly lethal to their species.
But they don't die.
Nope. These specific fish just keep swimming. They survive.
You know, it feels like magic when you look at it from the outside. You see these macroscopic, life-changing traits, and you intuitively assume there must be some massive visible cause.
Right, like something environmental or a huge developmental shift.
Exactly. But the reality is completely counterintuitive.
And that is exactly what we are mapping out today on this deep dive. We are tracing how a tiny, invisible structural change at the microscopic level, something so small you could never see it with the naked eye, creates a biological echo.
A really profound echo.
Yeah, we're going to follow that echo all the way up through a living system until it completely changes a breathing, moving organism.
It really is a profound chain reaction. There is a very specific pathway that governs this entire process in biology.
Which is what exactly?
We are moving from a change in a gene, which alters a protein, which shifts a physical structure or function, which ultimately manifests as a brand new trait. So, gene to protein to structure to trait.
Okay, let's unpack this. It sounds so clean and linear when you say it like that. But before we can even talk about how a mutation changes an organism, I feel like we need to understand how traits are built in the first place.
That's a good point. We need the baseline.
Right. Like right now, as you are listening to this, your body is expressing traits.
Yeah.
What is that baseline? How does a gene actually turn into a physical thing?
Well, let's start with the architecture inside your cells. The baseline involves two main players, the blueprint and the builders.
Okay.
The blueprint is your genetic information. Your genes are physical segments of incredibly long, tightly coiled DNA molecules.
And those sit inside the cells.
Exactly. Those DNA molecules are bundled into chromosomes, which sit securely inside the nucleus of your cells.
I've always struggled to visualize that scale, honestly, because words like DNA and chromosome and gene, they tend to just blur together for me.
Oh, absolutely. They do for a lot of people.
I heard an analogy once about nested physical scaling, comparing it to a book. Like, if the chromosome is the entire instruction manual, is the gene like a single chapter?
It's more like a single, highly specific sentence inside that manual.
Just a sentence.
Yeah. If you have this massive, heavy instruction book, which is the chromosome, the gene is just one sentence that gives a very precise command.
Okay. And the DNA itself?
That is the physical ink and paper making up the letters of the words.
I love that. So the DNA is the medium, the gene is the message, and the chromosome is the binding holding it all together.
You got it perfectly.
But, you know, sentences don't build houses. I mean, you could have a manual sitting on a desk, but it won't spontaneously assemble a table.
Right. Which brings us to the builders. The genes don't do any physical labor at all.
So who does?
The actual real-world execution of that genetic instruction is carried out by proteins. Proteins are the structural engineers, the bricklayers, and the machines of your body.
So they're the ones doing the heavy lifting.
Exactly. They form physical scaffolding, they transport oxygen, they digest food. And the wild part is they're constructed from different combinations of just 23 different amino acids.
Just 23 building blocks, making up all the complexity of life.
Just 20.
That is wild. But here's something that really threw me when I was looking into the sources for this deep dive. Every single somatic cell in an organism, so like a skin cell on your arm, a muscle cell in your heart, a neuron in your brain, they all carry the exact same master instruction manual.
Yep, the exact same book.
I always assumed my skin cells only had, skin genes, and my heart cells only had heart genes.
It's an incredibly common assumption. And logically, it makes sense to think specialized cells only hold specialized instructions.
Right.
But the reality is far more elegant. Every single cell in your body contains the entire book. Your skin cells contain the exact instructions for building a pumping heart ventricle or retinal eye structure.
Wait, then why isn't my arm growing an eyeball?
Because of gene expression.
Gene expression.
Yeah, it's not about what instructions are in the building, but which sentences are actively being read. In a skin cell, the chapter on how to build a heart is essentially glued shut.
Oh, I see.
The chemical machinery bypasses it entirely. The only genes that are turned on, the only sentences being read and handed to the protein builders are the ones relevant to being skin.
Okay, so the active genes dictate which proteins get built. But how does that actually play out mechanically?
What do you mean?
Well, we know proteins are the builders, but how does the shape of a protein dictate what an organism looks like or how it behaves?
It all comes down to a foundational rule in biology: structure dictates function. A protein is not just a uniform blob.
What does it look like then?
When those amino acids string together, they fold into a highly specific, complex, three-dimensional shape. Some parts repel water, some parts attract it, and they twist into these intricate origami structures.
Origami structures, that's a great image.
Right. And that precise 3D shape determines exactly how that protein locks together with other molecules in the cell.
Let's ground this with one of the examples from our research.
Wow.
Think about Darwin's bark spider.
Oh, the spider silk example. Perfect.
Yeah. These spiders can inherit different versions of a gene. Scientists call these variations alleles from their parents. And these different alleles act as slightly different sentences.
Slightly different instructions for building silk proteins.
Exactly.
And those tiny instructional differences alter the final origami fold of the silk protein. Let's say the gene instructs the cell to build a protein with a lot of molecular connectors.
Like little chemical hooks.
Yes, little chemical hooks on the outside of the folded shape. That protein is going to bind very tightly to the other proteins around it. They lock together aggressively.
And the macro result of that microscopic locking is a silk strand that has very low flexibility.
Right, it's rigid.
It's incredibly strong, but it refuses to stretch. But if the spider inherits a gene version that instructs for a protein with fewer of those connectors, the proteins don't bind as tightly. They have more slip.
Exactly, the web becomes stretchier.
Wait, I'm stuck here. I really want to push back on this to make sure I am visualizing the physics correct.
Sure, go ahead.
Wait, so a trait isn't just magically handed down. It's essentially microscopic Legos. If a protein is literally missing a connector bump, the whole web changes. Shouldn't the web just fall apart?
I get why you'd think that.
Yeah. How does a broken protein translate to a web that is stretchy and actually useful?
Well, let's look at it like a suspension bridge versus a solid concrete beam.
Okay.
If you have maximum connectors, that's the concrete beam, all the molecules are rigidly cross-linked. It can bear a lot of static weight, but if the wind blows too hard, it snaps.
Oh, because it can't bend.
Exactly. By removing some of those rigid crosslinks, you introduce physical space and slack between the molecules. They can slide past one another without letting go entirely.
Wow.
You haven't broken the protein, you've changed its tensile properties. It now absorbs kinetic energy by stretching instead of snapping.
That is so cool. It really is basically microscopic Legos.
Mm.
The physical shape of the brick determines the structural integrity of the entire castle.
And if a gene gets fundamentally scrambled, it might instruct for a protein that is completely non-functional.
Like it just doesn't work at all.
Right. It might lack all its connectors, or its 3D fold might be so unstable that it just unravels inside the cell. When that happens, the builder goes on strike. The job simply doesn't get done.
That makes the mechanics of it so clear. So we've established how normal baseline traits are built from these protein connections.
Baseline, yes.
But how do brand new traits suddenly appear in a population?
Yeah.
If an entire species is just passing down variations of the same Lego pieces, where does a totally new weird piece come from?
This is where we step into the territory of mutations.
Yeah.
What's fascinating here is how pop culture has done biology a massive disservice. The moment you hear the word mutation, your brain likely goes straight to science fiction.
Oh, absolutely. Radioactive ooze, comic book superheroes, giant monsters.
Right. We are conditioned to think of a mutation as a dramatic, overnight physical transformation.
Yeah, the idea that you touch something glowing and suddenly you can shoot lasers from your eyes or climb walls.
Exactly. But the scientific reality is far more mundane and, well, in a way, far more beautiful. A mutation is simply a structural, random chemical change to the genetic information.
Like a molecular typo.
Exactly. It is a molecular typo that happens during DNA replication when a cell is dividing to grow or to reproduce. The cellular machinery is copying 3 billion letters of code, and occasionally it inserts an A instead of a G.
That's it.
That's it, a typo in the manual.
And the crazy part is that right now, sitting here, you and I both have hundreds of these typos in our own bodies.
We do.
Because most mutations are actually silent or neutral. They happen in the non-coding parts of our DNA.
The blank spaces of the instruction book.
Right, the margins where no actual instructions are written. So
Meh.
the typo happens, but the protein builders never even read that section. Nothing changes.
And even when a mutation does occur in an active coding region, we have to address another major misconception.
Oh, the teleological misconception.
Yes, it's the completely natural but entirely false human assumption that organisms mutate because they need to adapt to their environment.
Right. The idea that a salmon swims into a dangerously warm patch of water, its body realizes it's overheating, and it somehow decides to mutate a new protein to survive.
Nature does not take requests. A salmon doesn't decide to mutate. A mutation does not happen in response to an environmental need.
It's just random.
Totally random. To truly prove this, we can look back at the historical experiments of Hermann Muller. He was a geneticist who worked with X-rays.
Okay, what did he find?
He demonstrated that if you expose an organism to radiation, it drastically increases the mutation rate.
But how does radiation actually do that? Does it, like, melt the DNA?
No, it's pure kinetic chemistry. An X-ray is a high-energy photon. When that photon passes through a cell, it physically collides with the atoms making up the DNA's sugar phosphate backbone.
It physically hits it?
Yes, it literally knocks electrons out of orbit, which breaks the chemical bonds holding the DNA strand together. The cell's repair proteins rush in to fix the break, but sometimes they put the wrong molecular letter back in the hole. That mistake is the mutation.
So it's an accident.
It is a random chemical accident governed by physics without any regard whatsoever for what the organism actually needs to survive.
It's like spilling coffee on an architectural blueprint.
Yeah.
The coffee doesn't know you're trying to build a structurally sound roof. It just randomly blurs a line.
That's a perfect way to put it.
And if the builders read that blurred line and put a window where a load-bearing pillar should be, the house collapses.
Exactly.
So if these are just random accidents, what actually happens when a typo does alter a protein's 3D shape?
This raises an important question about how we classify the biological outcome, and we have to be careful here because we are so used to labeling mutations as inherently bad or dangerous.
Right, like we said earlier.
But in biology, a change is just a change. We classify the effects of mutations based solely on how they impact the organism's ability to function and survive. They fall into three categories: neutral, harmful, or beneficial.
Let's start with neutral. The neutral ones are fascinating. Take the blue lobster.
Oh, I love the blue lobster.
Right. Normally, lobsters are a muddy brown or reddish color, but occasionally a random typo happens in the gene that controls a specific pigment-binding protein.
Astaxanthin, right, that's the pigment molecule.
Exactly. The mutated protein folds slightly differently. It still binds the pigment, but because the 3D shape is warped, it twists the pigment molecule.
It physically twists it.
Yeah, and that physical twist changes how the molecule absorbs and reflects light, turning the lobster's shell a brilliant neon blue.
But beneath the surface?
The lobster digests food fine, its shell is just as hard, and under current ocean conditions, being blue doesn't meaningfully impact its survival. It's just a cosmetic typo.
The paint color on the house changed, but the plumbing still works.
Exactly. Now let's contrast that with a harmful mutation.
Well, there is a documented genetic condition in some sled dog breeds that causes laryngeal paralysis.
Oh, I've heard of this.
Yeah. In this scenario, a random mutation alters the instructions for a structural protein that is vital for the peripheral nervous system.
So it's a nerve issue, not a throat issue.
Well, it starts as a nerve issue. The mutated gene builds a misfolded protein in the long nerves that run from the brain to the voice box, or larynx.
Right.
Because the protein is the wrong shape, the nerve fibers slowly degrade over time. Eventually, the electrical signal from the brain can't reach the muscles of the larynx.
So what happens to the dog?
When the dog exercises and tries to take a deep breath, the muscles fail to pull the airway open. The dog can suffocate.
Oh, that's terrible.
It is. That molecular typo broke a critical load-bearing wall. It drastically reduces the organism's ability to function and survive.
Wow. But then we get to the flip side. Here's where it gets really interesting.
The beneficial mutations.
Yes. Let's return to the Yukon River salmon we talked about at the very top of the deep dive. A random typo occurred in the DNA of a salmon. It altered a protein, and suddenly, the cell began producing a higher quantity of what are called heat shock proteins.
Right. And to understand why that is beneficial, you have to look at what heat actually does to a cell.
What does it do?
We talked about proteins folding into specific 3D shapes. Well, if the temperature gets too high, those proteins literally melt.
They melt.
Yeah. The heat energy breaks the delicate chemical bonds, and the proteins unfold, losing their shape and their function. It's exactly what happens when you fry an egg. The key proteins unfold and tangle together into a solid white mass.
So how do the heat shock proteins stop the fish from turning into a fried egg?
They act as molecular scaffolding.
Hmm.
The mutated fish produces these heat shock proteins, which physically wrap around the salmon's other vital proteins.
Like a shield.
Like a cage, really. They cage them in, preventing them from unfolding even when the water temperature spikes. The fish survives boiling waters because its microscopic architecture is reinforced against the heat.
That is incredible. But, it's crucial to remember the scale of how this rolls out in nature.
What do you mean?
A mutation like that affects a single individual first. It's not like the entire river of salmon collectively mutated heat armor all at once. It was a single typo in one isolated fish.
Exactly. Which brings us to the ultimate filter of biology.
The environment.
Yes, we've seen that a mutation causes a physical change. A twisted pigment, a degraded nerve, a molecular heat cage. But what dictates whether that new physical trait becomes the dominant feature of an entire species or dies out entirely?
It's the environment.
Yeah.
The environment is the final judge. So what does this all mean?
It means context is everything. There's no such thing as a universally good or perfect trait.
That's a really good point.
Let's think about thick fur.
Mhm.
If you are an animal living in the snowy Arctic, thick fur is a highly beneficial trait. It retains heat.
Right.
But if the global climate warms or you migrate to a desert, that exact same fur becomes a deadly hazard. The trait itself didn't change, the environmental context around it did.
There is a brilliant example of this with rough-skinned newts in Oregon State Park.
Oh, I know this one.
Yeah. If you look back historically, say, 200 generations ago, the newts in this population were mildly toxic, but none of them had what researchers might call a level 10 extreme poison trait.
They just didn't have the genetics for it.
Right. But today, if you go to that park, level 10 extreme toxicity is the most common trait in the entire population.
So how do we bridge that gap?
Exactly.
It starts with the typo. A random mutation introduced that extreme poison trait into just one newt, or maybe a small clutch of offspring.
But the mutation alone isn't enough to change the whole population.
No, it's not. The environment has to act as a selective pressure. In this case, the environment included their main predator, the garter snake.
It's an evolutionary arms race.
Mm-hmm.
The snakes are hunting the newts. The snakes easily eat the newts with the lower baseline levels of poison.
And those baseline newts die before they can reproduce.
Right. But the offspring that inherited the random level 10 mutation, they are too toxic.
The snake takes a bite, gets sick, and spits them out.
Yeah. So the highly toxic newts live longer, they mate, and they pass that specific mutated sentence in their DNA down to the next generation.
Over decades, the environment mechanically filters out the weaker trait and amplifies the mutated one.
It is a blind, unthinking sorting process.
It really is. And if we connect this to the bigger picture, because this entire cascade from a chemical bond breaking in DNA to a protein folding differently, to a snake spitting out a newt begins on a scale we absolutely cannot see, modern science relies heavily on advanced modeling to prove it.
And we aren't just talking about drawing simple arrows on a chalkboard here.
Not at all. Today, geneticists use cryogenic electron microscopy and computational biology tools like AI protein folding software to map the invisible.
So they can actually see it.
In a way, yes. We can take the wild-type genetic sequence, the normal baseline, and model the exact 3D shape of the protein it produces. Then, we input the mutated sequence.
And see the difference.
Exactly. The software shows us exactly how a single swapped amino acid changes the physical angle of a molecular connector.
It really removes the magic from the equation entirely.
It does!
You aren't just looking at a spider web and guessing why it's stretchy. You are using lab tools to point to the exact nanometer where a chemical hook is missing. And then you are mathematically calculating how that missing hook changes the tensile strength of the silk strand.
Biologists call it building a system chain. It proves causality.
Cause and effect.
Right. It confirms that biology is a rigid, mechanical chain of cause and effect. A chemical typo alters the 3D geometry of a microscopic builder, which alters the physical structure of a cell, which changes the macroscopic capability of the organism.
Let's pull all of this together for everyone listening, because we covered a tremendous amount of ground today.
We really did.
We started by looking at the master blueprint inside every single cell in your body. We zoomed in to watch a random, physics-driven typo alter a single sentence of that code.
The mutation.
Right. We watched that typo instruct your cellular builders to construct a differently shaped protein.
Which then changed its function.
Yeah, we saw how that slightly altered shape, whether it's a broken nerve or a new molecular cage, changed a physical function in the body. And finally, we watched the brutal reality of the environment decide whether that new trait would survive to be passed down or if it would be filtered out by natural selection.
It is a continuous, beautiful, and utterly relentless engine of biological variation.
It really is. So the next time you think about that sled dog running effortlessly through the heat, or you see a spider spinning a web that perfectly flexes in the wind, or you imagine those salmon pushing through boiling river waters, remember that none of those biological miracles started with a conscious choice.
They didn't occur because the animal tried hard enough or needed to change.
No, they started as entirely random, invisible chemical accidents in the dark space of a single cell.
When does a small change stay small, and when can it echo through an entire organism?